The idea I'm going to present today or to pitch today, that's on behalf of a full team, and Gabriele and Joachim are the other members of that team, and that's about a subtherapeutic, easy-to-use transporter cocktail. And what we want to achieve with that subtherapeutic transporter cocktail is to prevent unnecessary conduct of additional phase 1 studies during early development of new drugs, new chemical entities, and by that, of course, we will want to reduce the number of exposed healthy volunteers, the ecological footprint, but also kind of realize substantial cost savings. And to give you a bit background, so membrane, and so it's a transporter cocktail. So membrane drug transporters, they are located in many barrier tissues of the body. So for instance, the gut, the brain, the liver, the kidney, and they can be the cause of clinical relevant drug-drug interactions. So you see it below here in the small picture. So basically those drug transporters, they transport a drug outside of a cell or inside of a cell against the concentration gradient. So it's using energy essentially. And if you basically have two drugs, the one is transported and the other one is inhibiting or inducing a transporter, then this leads to a drug-drug interaction. Now, early identification of a new compound being such an inhibitor or inducer of a drug transporter is very important to avoid clinical relevant altered exposures of a co-administered drug. And actually, it's also requested by regulatory authorities. And what you typically do is you start with a first screen in a preclinical setting. And if you see there a relevant risk for drug interaction, then you would test that in a clinical interaction study with a sensitive probe substrate for the relevant transporter. Now, how is that done? So, and the classical development pathway to do that is first we would do a first-in-human study in healthy volunteers, which looks into the safety of a single-dose treatment and the pharmacokinetics of a new compound. That is followed by a multiple-dose escalation study, again in healthy volunteers, again safety and PK being the objective, and then we would enter the first patient study. That's the classical development pathway. Now, what is the earliest time point we could integrate or test for such a drug-drug interaction? And that is only because we are testing the two drugs both at therapeutic doses. That is only once we have the safety data from the new compound available. Because we cannot initially combine it, because then we would have mixed safety signals, and those initial studies are really for determining the safety of the new drug on its own. So that basically means if we have a single dose drug-drug interaction study, then the earliest time point would be once we have the safety data from this first-in-human study available. If we would do or need a multiple dose DDI study, then the earliest time point is after the multiple dose escalation study. Now, these are additional studies, and for cost reasons, then you often only test one dose of your new drug as a perpetrator, which, however, has some substantial uncertainty as the therapeutic doses at that time not known. Now, DDI data are relevant for patient studies, as those patients, and that's the next study here, they may take several co-medications. And with that approach, with that current approach, often we get the drug-drug interaction data too late to inform recommendations for the first patient study. Now, what do we suggest? And that is a so-called subtherapeutic, easy-to-use transporter cocktail. And what we want to do is to use a cocktail of different probe substrates at subtherapeutic doses. And using the subtherapeutic doses allows integration into first-in-human or multiple-dose escalation study directly, because subtherapeutic doses don't come along with any safety concerns. So we are still able to identify the safety profile of your new drug on its own, because there is no effect of the subtherapeutic doses. And that's how it would look like. So it would be directly, now shown by this dark blue bar here, directly implemented either in the single or multiple dose study here. And the advantage is then we have no separate drug-drug interaction studies needed. So it's directly integrated in the dosing in the studies. with the before already mentioned advantages of cost savings, reducing the ecological footprint and the number of healthy volunteers exposed. An additional advantage is that this probe cocktail can be integrated at more than one dose level because you don't need additional study arms, you don't need additional studies. So it's it's very cost effective to integrate it into one of those doses here, and it can be done at 2 or 3 doses without any large effect. And in addition, data will be available before the first study in patients and therefore will inform this first study in patient. Now, which data are available to support our approach? So there is already a transporter cocktail validated by Boehringer Ingelheim. And within that transporter cocktail, Boehringer, they used therapeutic doses of probe substrates. So the downside, as you can imagine, is again, it requires separate studies. So in principle that you can use cocktails to identify drug interactions, that is shown. But with that therapeutic dose cocktail, it still requires separate studies, and therefore we cannot realize the benefits I described before. And you have the publications related to that Boehringer cocktail here. There's another very interesting cocktail validated by Merck, and that transporter cocktail uses microdoses in the microgram area. We have checked that cocktail as well, and implementation in principle in early studies is possible. However, the preparation of the probe compounds at microdose levels is very laborsome from the CMC perspective. So it requires additional CMC work. You can order this GMP material, which you need, only usually at kilogram or even ton scales. And you need basically very small amounts only. And this altogether needs a lot of lead time before each drug-drug interaction study or before each first in human study, multiple dose escalation study, and thus it isn't used. Now, the idea of the subtherapeutic transporter cocktail is to use available market formulations. So, for instance, pediatric solutions that are then prepared at small amounts. So you basically, instead of the typical 3 milliliters you use for pediatric solution, you just administer 0.3 milliliter, but you can do that directly from the marketed formulation. And that can be directly prepared and conducted by the site conducting the first in human or multiple dose escalation study. So with that, we realized basically a cocktail that can be integrated immediately in the first in human or multiple dose studies without any additional efforts prior to the study in preparing the cocktail, and the cocktail is directly prepared by the site. So what needs to be done to implement the subtherapeutic easy-to-use transporter cocktail? So a clinical validation study is needed. As you have seen it was done for the Boehringer cocktail, it was done for the Merck cocktail, and what you there typically do is you test the effect of prototypical inhibitors and inducers on the probe substrates that are in the cocktail, and show that the effect is of the same magnitude as you would expect at full doses. This validation study for the subtherapeutic cocktail will also test the technical feasibility of cocktail preparation. We consider the likelihood of success very high, given that the therapeutic dose cocktail has been already validated and a microdose cocktail has been validated. We already looked into the selection of probe substrates and marketed products, and we have already concluded that a clinical validation study has been designed and the protocol submitted to German authorities. And now we are looking for funding collaboration for the DDI study conduct and evaluation, including activities like bioanalytics and so forth. And I won't go into that slide. That's essentially the graphical summary. So what is the current approach? What is the approach we are envisaging once the validation study is final? And with that, I'm already at the end of the presentation. I'm happy to go into the discussion. I will stop sharing here.